Well, welcome back, everyone. We're about to tackle the sixth lecture in the kidney series, and today we're going to focus on potassium. Of course, we started off talking about body fluid compartments, the delivery of plasma to the kidney, where the glomerular filtration rate enabled the tubular epithelium to reabsorb sodium and water based on principles of tubular transport. And we talked about the regulation of salt and water. Now we're going to return to potassium and to talk about how the kidney handles potassium and how indeed potassium is regulated. So we'll start off by beginning with some general comments about potassium, and then we'll go on to speak about the distribution of potassium. Of course, we've already talked about how most of the potassium that sits in our body fluid compartments is within the cells. And then we'll talk about nephron function. So we'll return to that picture of the nephron, and we'll return to how the kidney handles potassium. And then at the end, we'll look at how we regulate it. Now, this brings us back to the cartoon. These are our total body fluid compartments. We inverted, and here we recognize, of course, most of the potassium within our bodies resides in the intracellular fluid space, and yet each day we take in about 100 millimoles of potassium on average, and we have to excrete 100 millimoles of potassium in order to maintain potassium balance within the body fluid compartments, dominated by potassium intracellularly, but also the potassium that resides outside of our cells and how we regulate that. And so we're going to present the kidney with the glomerular filtrate, and then that glomerular filtrate is going to transit the nephron, and we're going to focus on the key role that the kidney plays in handling and regulating potassium excretion. And what's remarkable is we're going to see that that resides dominantly in the distal aspects of the nephron, particularly in the medullary and cortical collecting duct. So, case of the day. Previously healthy 18-year-old boy noticed a progressive weakness over several weeks, and he particularly noticed it in gym class when he had to be active and to go through exercises and run. On the day of admission, to hospital, he became unable to even stand after gym class, and he was brought to the emergency room. He looked entirely well, but he had severe muscle weakness. And what was remarkable was that his serum potassium concentration was profoundly low. The normal serum potassium concentration in you and I ranges between 3.5 and 5 millimoles per liter in the extracellular space, of course, much higher intracellularly. And the extracellular fluid concentration of potassium was only 1.2 millimoles per liter. So let's see if our understanding of kidney handling of potassium and potassium balance will allow us to gain some idea as to why this young man presented with such a profoundly low potassium, and why that presentation with such a profoundly low potassium was associated with clinical symptoms that didn't allow him to participate in regular physical activity. Well, first, a couple of things about potassium. So overall potassium balance, the typical North American diet contains 50 to 100 millimoles of potassium. Early humans may have eaten as many as 300 millimoles of potassium per day, that so-called paleo diet, and indeed our kidneys are likely to have evolved in a circumstance in which they played an important role in excreting potassium in order to maintain potassium balance or homeostasis in the face of that ongoing potassium load. Virtually 100% of what we present to the gut epithelium is reabsorbed, and of that, about 90% is excreted in the urine, the remaining small amount in the stool. And we're going to, for simplicity's sake, really look at the kidney as the, of course, dominant but almost the exclusive way in which you and I excrete the excess potassium that we take into our bodies each day. Well, where does it come from? Well, that begins on that dietary potassium, fruit and vegetables, bananas, orange juice. And you can see this list of high potassium containing including milk. Raisins, for example, because raisins are just, as we talked about in water balance, you take a grape and you remove the water and you end up with a raisin. Of course, the constituents of that raisin, the solutes within that fluid, even though the volume changes, remain quite high. So this is where predominantly potassium comes within our diet. And where is most of it? Well, let's talk about the distribution of potassium, because that becomes a key factor in our understanding of how we maintain potassium balance. And here I've drawn a beaker. And in that beaker, just a single cell floating in an extracellular fluid, just for simplicity's sake. And we remind ourselves that within the extracellular fluid compartment, the potassium concentration is 4 millimoles per liter. That's from our very first lecture on body fluid compartments and the solute composition of those body fluid compartments. And if we look in the intracellular space, the potassium concentration is the mirror image, if you like, of the sodium concentration. It's 150 millimoles per liter in the intracellular fluid compartment. And so if we do the math and then take into account the volume, if we think, okay, there's 14 liters approximately within the extracellular fluid compartment, that means that the total amount of potassium in the extracellular space is 56 millimoles per liter, on average in a 70 kilogram person. And if there are 28 liters within the intracellular space, then 28 times 150 yields 4,200 millimoles in the intracellular space. So in you and I, the vast majority of the potassium resides within the intracellular space, and that's a function of the concentration, and it's a function of the volume. Only 2% or less is in the extracellular fluid space, but that's what the kidney is going to see in part. So one of the key things that we have to think about with potassium is how it contributes to the resting membrane potential of cells. And it's important to understand this because we're going to move forward to excitable cells that require the generation of a resting membrane potential and a change in that resting membrane potential that facilitates their cellular activity. Of course, that's dominated by neurons, but also by muscle cells. So a leak of potassium from the inside to the outside of a cell through potassium channels within the cell membrane serve to generate an interior of a cell that is negatively charged relative to the outside. So you can imagine in that beaker setting, you have a very high concentration of potassium within that cell. And there's a great concentration gradient favoring the movement of potassium to the outside of the cell, and to facilitate that movement, there are potassium channels. And so the high concentration drives the movement of potassium out of the cell, generating a negative electrical potential gradient. And the determinant of the transmembrane potential gradient is a function of the ratio between the extracellular fluid potassium concentration and the intracellular fluid potassium concentration. And the resting membrane potential is critical for muscle, nerve, and of course cardiac function. This is the Nernst equation. It's meant to show that that potential gradient is a function of the potassium concentration inside and outside of the cell. So let's go back, and so we can see, well, indeed, actually, that potassium concentration so very high inside of the cell relative to outside of the cell drives potassium out through these potassium channels, generating an intracellular negativity relative to the extracellular space. So what other factors then? So sitting there, that cell, unperturbed, but sitting with a high intracellular potassium concentration and a low extracellular potassium concentration, has a concentration gradient driving the movement of potassium from in to out of the cell that renders the cell with a potential gradient and a negativity within the cell. What other factors? Well, this model demonstrates that there are two important hormonal mechanisms that influence the potassium distribution, other than, of course, sodium potassium ATPase. So this is meant to represent here a muscle cell. Now, of course, sodium potassium ATPase moves potassium into the cell and sodium out of the cell so that there's a low sodium concentration and a very high potassium concentration in the cell. Now, one of the things that influences the movement of potassium in and out of the cell that converge, if you like, on the activity of sodium potassium ATPase are two receptor systems. The first, and perhaps the most important day-to-day, is in fact the insulin receptor. Cartooned here is an inverted Y. And this hexagon is meant to represent insulin. When insulin, as the ligand, engages the insulin receptor, cell signaling generates activation of PI3 kinase and AKT, and that leads to the movement of glucose into the cells. That's what you and I depend on when we eat sugar. However, another function downstream of this receptor is the activation of atypical protein kinase C, a kinase in the cell that phosphorylates sodium potassium ATPase and increases its activity. So although you and I use insulin to move glucose into cells, a consequence of insulin signaling in a muscle cell is to activate and drive an increase in activity of sodium potassium ATPase. So insulin will shift potassium from the inside to the outside of the cell. It'll cause a relatively small perturbation because remember, in you and I, only 56 millimoles of potassium is out here, and 4,200, is inside the cell. So insulin shifts the equilibrium and moves potassium into cells. The other receptor system is the beta-2 adrenergic receptors. Now, insulin receptors are tyrosine kinase inhibitors, and beta-2 adrenergic receptors are G-protein coupled receptors that generate cyclic AMP and activate another kinase called protein kinase A. And therefore, if I give a beta-2 catecholamine that engages the receptor, I will increase the cellular activity of protein kinase A, and it will phosphorylate sodium potassium ATPase and increase its activity. So if I want to shift the balance between intracellular and extracellular potassium, I can drive potassium into the cell by taking into account these two ligand receptor systems. Now, in hospital settings where we have emergencies associated with very high potassium levels, we use insulin as a treatment, and we use beta-2 catecholamines as treatments to drive the potassium into the cells, because that can have an important effect on the transmembrane electrical gradient and reduce that gradient in settings that allow muscle function to be normal. So insulin promotes potassium uptake, and beta-2 catecholamines promote potassium uptake. They both converge on the activity of sodium potassium ATPase, which serves to push potassium into cells and extrude sodium from those cells. Acidosis is another important factor. So remember, a muscle cell, again cartooned here, has the ubiquitous sodium-hydrogen exchanger, and this functions down concentration gradients. So sodium moves into the cell, hydrogen ion moves out of the cell. It's the sodium gradient and the hydrogen ion gradient that drive the activation. Now imagine a situation in which you have a high concentration of hydrogen ion outside the cell. That diminishes the gradient for hydrogen to move from the inside to the outside of the cell. And what that does is it reduces the entry of sodium. Now when sodium enters, it's pumped out by sodium potassium ATPase, and potassium enters. So acidosis, a high hydrogen ion concentration, reduces sodium hydrogen exchange, reduces entry of sodium into the cell, reduces the movement of sodium out of the cell via sodium potassium ATPase, and limits the entry by limiting the activity of sodium, potassium, ATPase. So the higher the hydrogen ion concentration in the surrounding extracellular space, the less able a cell is to take up potassium. So what's the effect? Well, the effect of shown in this cartoon, and I don't want you to pay attention to the other transporters, but really focus on the sodium hydrogen exchanger and on the sodium potassium ATPase here. And reduced activity of the sodium hydrogen exchanger by virtue of a high hydrogen ion concentration will reduce the activity of sodium potassium ATPase and tend to subtly shift the equilibrium between potassium in the cell and potassium outside of the cell. So one of the things we might ask ourselves is we understand that potassium is high in cells because of sodium potassium ATPase activity. But there are other factors that influence the activity of sodium potassium ATPase, and that those factors serve to cause subtle adjustments in the distribution of potassium from inside to outside of the cell. Beta-2 catecholamines and insulin drive activity of sodium potassium ATPase, serving to shift potassium into cells. High hydrogen ion concentration tends to blunt the activity and prevents potassium entry into cells. What are the consequences? Well, this is shown here in a membrane potential. So this is the extracellular potassium concentration in a cell membrane potential. If you have low potassium concentrations outside of the cell, then that tends to hyperpolarize the cell. Why? Because it serves to increase even more so the gradient for potassium to leak through potassium channels from the inside to the outside of the cell. So a consequence of low potassium concentrations outside of the cell and high potassium concentrations inside of the cell is to hyperpolarize, to make it more difficult for that cell to reach a threshold that allows it to generate an action potential, for example, in a muscle cell. High potassium concentrations outside the cell serve to blunt the concentration gradient that drives the movement of potassium from inside to outside of the cell. That reduces the tendency of potassium to move out of the cell, reduces the tendency of that potassium movement to generate intracellular negative potential, and the cell depolarizes. So extracellular potassium concentrations, if they're low, they cause excitable tissue, muscle cells, nerves, heart cells to hyperpolarize. High potassium concentrations cause them to depolarize, and as a consequence affect the ability of that cell to generate an action potential and to influence its biological activity. So one of the things that we can see here, for example, is if in this beaker example again, here's our single cell. If the extracellular fluid concentration is just 2 millimoles per liter, then that serves to enhance potassium exiting the cell, serves to increase the negativity in that cell, and the cell is hyperpolarized. That low to outside the cell potassium to inside the cell potassium ratio is a direct determinant of that membrane potential difference. And in the opposite fashion, again our beaker, again our cell, a high extracellular potassium concentration serves to diminish the amount of potassium that transfers out of the cell. And as it diminishes the amount of potassium, as that outside potassium concentration rises, then we tend to have a less polarity, and so the membrane potential, the potential gradient across the cell membrane declines. Now, this is what are the clinical consequences. So let's look at hypokalemia on the left side. In hypokalemia, remember the cell is hyperpolarized, and look at the consequence is muscle weakness. It also promotes cardiac arrhythmias. So remember, our young patient presenting to the emergency department had a very low potassium concentration. What would that potassium concentration have done? Well, it would have hyperpolarized the cell and made it more difficult to generate an action potential, more difficult to contract and contribute to the symptom of weakness. And the EKG, this is a trace off a heart, and I know you've seen these in your cardiac physiology. It tends to get abnormal. It gets the peak wave, the T waves change, the ST segments change, and these changes in the ST segment particularly promote the development of ventricular arrhythmias, like ventricular tachycardia. And so arrhythmias of heart function are a consequence of low potassium, and low potassium influence skeletal muscle function by hyperpolarizing, making it more difficult to contract and contributing to weakness. Now, hyperkalemia does the opposite. It is associated with life-threatening cardiac arrhythmias, but here's the progression of here. This is the low potassium, very similar to here. Here's normal potassium, and as the potassium concentration rises, the EKG tends to flatten. It's like you took the EKG and you pulled it like a string, and all of a sudden you've got a very flat EKG. Now the next step in this is asystole, so that hyperkalemia causes electrical gradient differences in the heart reflected in the EKG that lead to eventually asystole, no contraction, cardiac arrest. Hypokalemia can cause ventricular tachycardia, which also can cause cardiac arrest. So you get an abnormal EKG, but it also causes muscle stiffness and weakness because it just hyperpolarizes the cell, hypopolarizes the cell, and as a consequence, there is an inability to generate that action potential that supports the ability of that muscle to contract. The muscle feels rather stiff and tired. So one of the interesting things is that the importance of maintaining potassium within a normal range is that to have it too high is to influence the electrical potential across cell membranes and to make them anergic and stiff. To have low potassium is to make them weak, unable to generate a contraction, and to cause EKG changes that promote the development of arrhythmias. Clearly, of course, maintaining that potassium within a normal range becomes important. So a couple more things to remember here. So when you eat, so a high potassium meal could have, depending on how many fruits and vegetables you consume, could have 60 millimoles of potassium. Now that potassium goes into the extracellular space. And if you think that the extracellular fluid volume is only 14 liters, with a concentration of potassium of 4 millimoles per liter, that's 56 millimoles of potassium. So if you ate 60 millimoles of potassium in a single meal and it just sat in the extracellular space, then in fact you'd have a big problem because you double the concentration of potassium. It would go from 4 to 8, you would have a cardiac asystole, and you would die. And that's the cardiac arrest. Well, you don't die when you eat a high potassium meal because that comes in fruit and vegetables that contain glucose, and that leads to insulin secretion. What does insulin do? Well, remember, it's one of those 3 factors that plays a key role in the distribution of potassium from the inside to the outside of the cell. Insulin engages its receptor, drives the activity of protein kinase C, phosphorylates sodium-potassium ATPase, increases its activity, and potassium is moved into the cells. Now, 60 millimoles of potassium into 28 liters with 150 millimoles of potassium causes a trivial change in the intracellular potassium concentration. So you and I can eat a meal high in potassium, high in vegetables and fruit, and not suffer a cardiac arrest because our physiologic response, engaging insulin, drives that potassium meal into the cells. But then eventually, you and I are going to have to excrete that. When you exercise, the contraction of the muscles leads to a release of potassium. It leaks out of the cells. And the local increase in potassium is quite important because it causes arterioles to dilate and helps maintain muscle blood flow. Now that's critically important because the increased muscle activity will require more oxygen delivery and increase in blood flow, so that the vasodilating effect of the potassium release in muscle has a important physiological consequence. The excess potassium, though, in the extracellular fluid space can have deleterious effects, though, on the muscle and on the heart and on nerve function. And so the adrenaline that's released as a catecholamine serves to engage those beta-2 adrenergic receptors on the cell membrane and redistribute the potassium back into the blood. So that exercise may be associated with an acute rise in potassium, facilitating blood flow, and then you redistribute that potassium back into the cells as a consequence of that beta-2 agonist activation of the receptor, activating protein kinase A, increasing sodium-potassium ATPase activity, and moving potassium from outside to the inside of the cell. So now, of course, if you and I sit there and we keep eating bananas and fruit and we keep moving potassium from the extracellular fluid into the intracellular fluid, eventually what we're going to be faced with is increasing our total body potassium. And we're going to have to figure out how to get rid of it. And that's where the kidney comes in. The kidney plays a key role in maintaining normal potassium by facilitating its excretion. So you can imagine, you eat those bananas, it goes into the extracellular space, it's shifted into the cells, and then it's secreted by the kidney so that you maintain overall homeostasis or balance of potassium in your total body water. So let's talk about nephron function and potassium and see how we can look at the kidney and where in the kidney is potassium handled. Now, here's our nephron, our proximal tubule, our loop of Henle, our distal tubule, and our collecting duct. And we're going to look at the key effects in the next several slides on how these segments influence potassium. I showed you a long time, or one of the earlier lectures on tubular function, that potassium is reabsorbed in the proximal tubule. And it tends to be reabsorbed in a manner that's shown here, which might be surprising. So here's a lumen, here's a proximal tubule cell shown here, and here's the blood compartment or the peritubular capillaries on the other side. Potassium is small. It's so that if you and I are sitting in the lumen of a glomerular capillary, the potassium concentration is 4 millimoles per liter. And if you and I are sitting in Bowman space in the glomerular ultrafiltrate, the potassium is 4 millimoles per liter. It moves unencumbered across the glomerular capillary wall as water moves across the glomerular capillary wall. And remember, water moves across that capillary wall in response to Starling forces. Most of the reabsorption of potassium in the proximal tubule is due to solvent drag and paracellular diffusion as water is reabsorbed. So remember, sodium reabsorption and extrusion into the peritubular space drives the reabsorption of water across the cell, but also between the cell. It's a leaky capillary. So as the proximal tubule transports sodium and moves it into the peritubular space, water follows that. And as the water moves, it drags that small solvent, potassium, with it. So much of the potassium that's filtered by the glomerulus into Bowman space is reabsorbed through a paracellular diffusion or solvent drag as water is reabsorbed in the proximal tubule. Now, the next thing that happens is that the potassium is reabsorbed within the thick ascending limb. Now remember, the thick ascending limb has a famous transporter, the sodium-potassium 2-chloride transporter. And that accounts in part for potassium reabsorption. I'm going to show you what the consequence of that is. So if you and I enter Bowman space and we ride through the nephron, looking around at us, what are we going to see? Well, we're going to see a lot of potassium disappear by the time we get to the end of the proximal tubule. And then as we zip down the loop of Henle and come up the thick ascending limb, all of a sudden the potassium is going to start to leave again because sodium potassium 2 chloride transporters will shift it out of the lumen into the cell where it'll be pumped out, where it will be transported out into the peritubular space. But also there'll be some paracellular movement. I'll show you that in a second. So that if you and I arrive up here at the start of the distal convoluted tubule, and particularly as we get to the collecting duct, there's very little potassium around. So one of the interesting things about the handling of potassium by the kidney nephron is that proximal reabsorption of potassium reabsorption of potassium in the proximal renal tubule and in the ascending limb of the thick ascending limb of the loop of Henle, virtually it reabsorbs all the potassium filtered. Now, you're going to want to have potassium at the end of the day to excrete, and that tells us that all of the potassium that we see in our urine is secreted in the collecting duct. And we're going to talk about that. So here's a thick ascending limb cell, and here's the lumen. Here are the cells here. Here's the peritubular blood compartment. Here's our famous sodium potassium 2 chloride transporter moving potassium into the cell. There's some recycling of potassium so that this transporter can continue to work. ROMK stands for a renal outer medullary potassium channel. So it's just a potassium channel that resides within the luminal membrane of the thick ascending limb cell. And as water reabsorption comes and as potassium is reabsorbed, particularly peritubular potassium transport occurs. So the thick ascending limb has a mechanism for reabsorbing and partially recycling potassium, and there's also the paracellular movement of potassium, really electrical chemical gradients driving that reabsorption. So the thick ascending limb mops up the potassium that remains within the lumen. So potassium gets out of the lumen in the proximal tubule and it's removed from the lumen in the thick ascending limb. So almost all the potassium that's filtered at the glomerulus as a consequence of Starling forces and water movement is reabsorbed. And that means that all of the potassium in the urine, which is by and large equivalent to what your daily intake is secreted in the collecting duct. So now we want to look at the collecting duct. We want to look pretty carefully at what are the factors that influence the ability of the collecting duct to secrete potassium. And so we're going to be talking about particular transporters that we've heard about before, the ENaC, the epithelial sodium channel. This is a channel that resides within the luminal membrane of the collecting duct that allows sodium entry down a concentration gradient into the lumen from the lumen into the cell. Importantly, that sodium movement through the epithelial sodium channel is driven by concentration gradients that are a function of sodium-potassium ATPase activity at the basolateral membrane, maintaining that low sodium concentration in the cell and driving reabsorption of sodium. There are lots of potassium channels in these cells, and I'll show you a closer cartoon of this, the renal outer medullary potassium channel, and another channel is called MAXI-K. In some physiology textbooks and in physiology research, this is referred to also as BK. MAXI, or B, the B stands for big potassium. These are very high capacity potassium channels. These are integral membrane proteins that reside within the luminal membrane and sometimes the basolateral membranes of cells that allow potassium to move into the cell, depending upon it allows potassium to move out of the cell more properly. So let's take a look at that. So here's our lumen. Here's our cortical collecting duct, or so-called principal cell, one of the two important cell types within the cortical collecting duct. And here's our basolateral membrane, our luminal membrane. Here's our old friends, so to speak, our sodium, potassium ATPase residing within that basolateral membrane. And here's our famous epithelial sodium channel that allows sodium to enter down a concentration gradient. And here's these luminal potassium channels that allow potassium to move primarily on the basis of concentration. Also, electrical gradients from the inside of the cell to the outside. So now we have in place a mechanism in which sodium entry goes down a concentration gradient and potassium exits. It's that positioning of those integral membrane proteins, these ROM-K and MAXI-K channels, that take potassium and allow it to re-enter the lumen. So now what you see is an important determinants of potassium secretion, the intracellular potassium concentration. The low tubular concentration of potassium, so high flow rates through the lumen of the nephron, lower the potassium concentration and enhance its ability to move outside. There are other transporters we're not going to really focus on. The key principle here is that there are integral proteins in the luminal membrane of the cortical collecting duct that facilitate the movement of potassium from a high concentration inside the cell to a low concentration outside the cell. Now, the last thing to note is that the cells tend to be relatively impermeant to chloride. There is a paracellular pathway so that when sodium is reabsorbed, the lumen becomes a little bit negative relative to the interior of the cell. And that means that a positively charged particle like potassium can move readily down what we would say a concentration and electrical gradient. So in the cortical collecting duct, potassium is secreted through potassium channels on the basis of a concentration and electrical gradient. And the electrical gradient is dependent on sodium reabsorption through the epithelial sodium channel. So you can imagine a situation in which if you want to encourage the cortical collecting duct cell to secrete potassium through these channels in the luminal membrane, you want to give it some sodium and you want to make sure that there's a low concentration of potassium. So high urine flow rates, sodium delivery, and the sodium epithelial channel are predeterminants of the passive secretion of potassium from the inside of the cortical collecting duct cell into the lumen. So the ability of the kidney to secrete potassium is a passive process that's a consequence of the activities of other channels and transporters in the basolateral and luminal membrane. Well, my scrolls are becoming a little bit unreadable here. There are other cells, and we'll come to these cells in the next lecture, which are the alpha intercalated cells, and these cells are responsible for secreting hydrogen ions. So I just point out that the two major populations of cells in the cortical collecting duct, the principal cells, which dominate their ability, are dominantly responsible for potassium secretion, and the intercalated cells, which play a key role in the secretion of hydrogen ion, which is an active process. So if you're sitting there in the cortical collecting duct, the ability to move hydrogen ion into that lumen requires energy, ATP. The ability to move potassium depends upon electrical and concentration gradients, and it is passive. All right, so let's talk about, those are the general. So here's the nephron, filters lots of potassium and then reabsorbs it all. And then it has a mechanism in the distal nephron in the cortical collecting duct that facilitates the passive secretion of potassium. And it's the regulation that we want to look at now that will tell us how much the potassium secretion will be, how much potassium secretion will be required to maintain potassium balance. So here are the factors that determine potassium secretion. The delivery of sodium. Why? Because sodium entering the cell through these epithelial sodium channel generates a lumen negativity that attracts positive to negative, facilitates the movement of potassium outside of the cell. Tubular flow rates. Well, the higher the flow rate in the tubule, the lower the potassium concentration. You can imagine that if there was no flow, you'd pump potassium into the lumen until the concentration's equilibrated. But if you have running water through that lumen, you put the potassium in, it flushes it away, and it keeps the concentration low. So sodium delivery and tubular flow rates. Lumen negativity, which is actually a consequence of sodium delivery. And then what we're going to come to, aldosterone, a hormone, a steroid hormone that has a key role in regulating potassium secretion. So where does aldosterone, where is it made? And how is it secreted in response to potassium? Well, two important things. It sits within the zona glomerulosa of the adrenal cortex. So the adrenal cortex, the adrenal glands sit on top of the kidneys. And in the cortex of the adrenal gland is a zone that's been named by the anatomist zona glomerulosa. It secretes aldosterone. What is a key stimulant of the secretion of aldosterone? Well, you probably don't have to guess what that is going to be. It's going to be potassium. So high potassium stimulates aldosterone secretion. Low potassium inhibits its secretion. So one of the ways in which the kidney regulates its ability to secrete potassium is by responding to aldosterone, which is secreted by cells in the adrenal cortex based on the potassium concentration that they see outside of them, likely based a little bit on membrane potential. So the question you and I now have is, okay, well, we sort of pushed the process down the road a little bit. The question is, if aldosterone is secreted, its secretion, if aldosterone secretion is regulated by potassium, then how does aldosterone serve to regulate potassium secretion? If high potassium drives aldosterone, then how does high aldosterone drive passive potassium secretion? And to do so, we're going to look at this cartoon here. Now, here's our cells in the cortical collecting duct. We're really going to be thinking them as principal cells in the cortical collecting duct. And this is our lumen, and this is our peritubular space with the blood compartment and the interstitial fluid compartment. So what happens to aldosterone? So aldosterone is cartooned as these little yellow circles. So it's a steroid hormone. So if there's a high potassium in the adrenal gland, will stimulate aldosterone, so its concentration will rise. The aldosterone will diffuse across the basolateral membrane of the cortical collecting duct principal cell. And it engages a receptor, a cytoplasmic receptor. So the paradigm here is a steroid hormone that diffuses through the lipid bilayer, enters the cell, binds to a receptor within the cytoplasm, and that ligand receptor combination translocates into the nucleus and influences gene transcription, becomes a transcription factor, an active transcription factor. Well, what are the consequences? So high potassium, high aldosterone enters the cell, it increases the synthesis of genes. Well, one of the genes that it causes to increase in expression is the epithelial sodium channel. So why would that be important? Well, if you have high potassium, you want to have lots of epithelial sodium channels to facilitate sodium entry. That drives the lumen negativity and helps the electrical gradient that support passive case secretion. The second thing it does is it puts potassium channels into that luminal membrane. So The cell sees aldosterone. It sees more, the response, more epithelial sodium channels, more potassium channels. So the permeability of potassium in that membrane is enhanced. So what's the consequence? More aldosterone, more sodium reabsorption, and more K secreted. The aldosterone drives lumen negativity. It drives the ability, and that drives the ability of that cell to move sodium in, and it drives the permeability and allows potassium to move down the concentration gradient. Remember, the more sodium that comes in, the more sodium exits down here. And when sodium exits, it keeps putting potassium in. So you start to drive the movement of sodium in, sodium out, potassium in, potassium out. So sodium in, sodium out, potassium in, potassium out. The consequence, if you like, of aldosterone and its effect on these transport proteins that all reside in the luminal membrane, the principal cell. So what is the action? Well, it has a number of other effects too. It actually increases the number and open probability of sodium channels. So that's the expression and the open probability, increasing sodium permeability. The more sodium you get in and then pump out, the more potassium you get in and then allow to move out of the cell, opposite membranes. It increases sodium reabsorption. These together combine to increase the lumen negativity, and they all combine to increase increase potassium secretion passively. So the question you might have is, how can one hormone regulate both sodium and potassium together? And then let's just walk through that. So how can we achieve potassium balance when, for example, there's a decrease in the extracellular fluid volume, which will drive the renin-angiotensin system? So a low volume state will reduce GFR, will reduce the flow rates within the nephron, the lumen of the nephron. So when you come to the cortical collecting duct, if extracellular fluid volume is low, the urine flow rates will be low. Low urine flow rates will serve to reduce passive K secretion. High aldosterone, as a consequence of that extracellular fluid, the decreased extracellular fluid volume, will serve to increase potassium. So what happens is you're in balance, you reduce passive K secretion because the flow rates go down, but you increase passive K secretion by virtue of the aldosterone. So you maintain balance by having those opposite effects of a rise in aldosterone and a reduction in the urine flow rates. How is potassium balance achieved? With the changes in effective circulating volume? So imagine potassium volume expansion, high salt intake, for example. The expanded high extracellular fluid volume will inhibit renin, inhibit the formation of angiotensin II, and reduce aldosterone secretion. The expanded extracellular fluid will raise the GFR, inhibit proximal reabsorption, and increase the flow rate to the collecting duct. So increasing flow rate will serve to increase passive K secretion, but the low aldosterone will tend to limit the passive K secretion so that you get this independent regulation of volume and potassium by having these counteracting influences. The other thing you'll recognize is two important things converge on the adrenal gland to cause an increase in potassium, in an increase in aldosterone secretion: high angiotensin II and high potassium levels. They're often at cross purposes, but these differences can show you how you maintain that balance. So what happened when you ate the banana? Well, this picture is a little bit complicated, but here's the banana, the increase in the potassium intake, and the potassium gets reabsorbed into the bloodstream. After the potassium, there's sequestration, so the potassium goes into the liver and the muscle. And that's the first thing that happens. So when you eat that potassium load, it's stored in tissue. The second thing that happens is indeed you cause changes in the handling of potassium within the kidney nephron. We've said that higher potassium will drive aldosterone. That will drive the loss of potassium. So you'll get potassium excretion. The other thing that I didn't focus on is in the distal convoluted tubule. High potassium inhibits sodium chloride cotransport. So one of the consequences of a high potassium inhibiting sodium chloride cotransport is it increases delivery of sodium to the collecting duct and increases delivery of water, less water reabsorption. And so high potassium sets the stage, if you like, for facilitating K secretion by increasing flow rates, increasing sodium delivery, and stimulating aldosterone. That's a perfect trifecta. That will drive K secretion so that you eat the potassium, you store it in cells, but the consequences of that, of subtle rises, it lead to secretion. So you move it into a storage and then gradually excrete it out, preventing wide excursions in the potassium concentration depending on what you eat. So kidney responses to potassium allow you to maintain balance after you store that potassium load within tissue. And that's illustrated here. So early on you store, later on you excrete, and those are consequences of things happening within the distal convoluted tubule and dominated by that biology we've just reviewed within the cortical collecting duct. So that brings us back to sort of our summary slide. Our fluid compartments, our inverted beaker, potassium sitting in the cells, potassium intake, the delivery of water and potassium to the nephron, filtration, reabsorption in the proximal tubule and the thick ascending limb, and then secretion, allowing that 100 milliosmoles to be excreted by the kidney at the end of the day. So back to the case. Where do we sit with that? Well, case of the day, paralyzed. So this patient was treated with intravenous and oral potassium and they recovered the strength. So that's interesting because you would predict that, right? If you become somehow profoundly hypokalemic, that'll influence the charge or the potential gradient across cell membranes, and in contractile tissue like skeletal muscle and heart, that'll influence its function. He recovered strength when his potassium balance was restored because the transmembrane potential gradient was restored when his potassium concentrations in the extracellular space were normalized relative to the intracellular. It's interesting because this individual was found to have a high urine potassium level. And so that's counterintuitive because we don't expect that he would have anything stimulating the passive secretion of potassium by his cortical collecting duct. It was turned out that he had loss of function in a gene encoding the sodium chloride transporter. So the recognition that his kidney's response to hypokalemia was inappropriate, he continued to passively secrete potassium even when he was hypokalemic. The loss of sodium in the urine led to high aldosterone levels and the increase in the potassium loss in his kidney. Now, the sodium chloride cotransporter we talked about when we talked about sodium handling in the different segments of the nephron. And the loss of function leads to increased sodium delivery to the cortical collecting duct and increased water delivery to the cortical collecting duct. The loss of salt and water reduces his intravascular volume and activates the renin-angiotensin system, leading to that perfect system that enhances passive K secretion out of keeping with his ability to regulate that potassium secretion in a normal way. The sodium-potassium 2-chloride transporter is shown here in the fluorescence. It's a luminal transporter. Here's a cartoon of it. It has several membrane-spanning segments and indeed domains within the cell that regulate its activity in an increasingly complicated manner. And so that's shown here. The The sodium-potassium ATPase in a distal convoluted cell is responsible for reabsorbing sodium down a concentration gradient, and it just pulls the chloride with it. So chloride's the passive partner here, and sodium concentration gradients are driving that activity of that cotransporter. So less activity here, more sodium loss. This mutation in the sodium chloride cotransporter is actually called Gittleman syndrome. And I have actually a patient in my practice with Gittleman syndrome, and we're constantly trying to replace their potassium so that they maintain normal potassium balance and don't get symptomatic. So summary. Dietary sodium typically equals the urinary potassium excretion. Signal to the kidney is a change in the potassium concentration. Increased potassium increases excretion. Decreased potassium will decrease secretion. 90% of the filtered, close to 100, is reabsorbed by the proximal tubule and the thick ascending limb of the loop of Henle. The chordal collecting duct regulates potassium secretion, enables you and I to maintain potassium homeostasis or balance by altering the activity of those transporters and influencing passive K secretion. Aldosterone playing a critical role in that. All right, so this brings us to the case of the day, the dialysis patient with diarrhea. And so we'll talk about acid-base balance in our next lecture. So thanks very much. Take care.